Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, I hope all of you are fine and safe at your home. I have prepared this lecture by consulting these three books which are provided on this slide. I have also uploaded all the full text of all the three books on the LMS page of your course. So if you want to study the detail of this topic or to enhance your knowledge, please go to the LMS page of recommended books and read from these books also. So, in this lecture, the learning goals include to study the expression systems to express the foreign proteins. Then, what type of expression systems are used? We will study about the bacterial system, yeast system, insect and mammalian cells, and then we will also do the comparison of all of these expression systems. In this lecture, we will study the systems which are used for expressing the recombinant protein. Why do we want to purify or we want to express the protein in the host organism? Because if we want to study the function of a protein or the protein has some importance in therapeutic importance or it has some medicinal importance, then it is very difficult to purify the protein from the original cell because it is along with all the other proteins and it will be in very minute amount. So this is what we have in nature, and for our purpose of study we want it is huge quantity. So this is the quantity that we want. That's why we want to express the recombinant protein in different hosts. So first we will see that the clone genes can be expressed to amplify the protein production. Frequently, the product of a clone gene rather than the gene itself is of primary interest, particularly when protein has commercial, therapeutic or research value. Biochemists mostly use purified protein for various purposes. For instance, if they want to elucidate the protein function or they want to study the reaction mechanisms, for instance to study the industrial enzymes which are used in various reactions, or they want to carry out the structure studies of protein, they want to identify the protein structure or the protein has some importance like it can be used as a vaccine, antigen or antibodies, or it has some therapeutic application. So to proceed towards the purifying the protein or to fulfill its purpose, the biochemist has to follow the following flow. Like first of all, it needs to get the information about the target gene and the vector that is being used to express the target gene, which is done by using the bioinformatic tools. And after target identification, and the biochemist moves towards cloning of the gene. After the confirmation of cloned plasmids, we mostly confirm that cloning is successful, we send our sample for sequencing or we use the restriction digestion. Then we move towards the protein expression test. First we do some pilot experiments, and after observing that protein is being expressed at very high rate, we do main experiment, and after protein expression we move toward the protein purification. When the protein is purified, then it is used for what purpose it was purified. We will take an example like we want to express a eukaryotic protein in a bacterium host. For this purpose, we know that the eukaryotic proteins or eukaryotic genes have surrounding sequences which are required for their transcription and regulation in the cell they are derived from. But these sequences do not function in bacteria because promoters, ribosomal binding sites, and additional regulatory sequences are the elements which are required for their controlled expression in bacterial cell. So for this purpose, appropriate bacterial regulatory sequences must be inserted in the vector DNA at the correct position relative to the eukaryotic gene. In some cases, the cloned genes are so efficiently expressed that their protein product represents 10% or more of the cellular protein. At these concentrations, some foreign proteins can kill the host cell, so expression of the cloned genes must be limited to a few hours before to plan to harvest the cells. So, the cloning vectors with the transcription and translation signals needed for the regulated expression of a cloned gene is known as the expression vector. And the rate of expression of a cloned gene is controlled by replacing the gene's normal promoter and regulatory sequences with more efficient and convenient versions supplied by the vector. So, we will see the structure of the expression vector. This is the structure of an expression vector which contains all the elements which are positioned near the unique restriction sites which is used for cloning, so that the genes inserted at the restriction site will be expressed from the regulated promoter elements. So here is a well characterized promoter and its regulatory elements. As you can see, the operator sequences which are positioned near the unique restriction site which will be used for cloning purpose. So this is known as the MCS. As we know, we have seen this site also in the cloning vectors, that the gene is inserted at the restriction sites and it will be expressed from the regulated promoter elements. Then some of the vectors also incorporate certain features, like they have the ribosome binding sites to enhance the translation of the messenger RNA derived from the gene, or a transcription terminator sequence. Every living organism has the capacity to express the genes in its genomic DNA. So any organism can serve as a host to express the protein from a different species, or we can say for heterologous expression. So almost every sort of organism has been used for this purpose, and each host type has a particular set of advantages and disadvantages. Here we will study about the bacterial expression system, yeast expression system, insect expression system, and mammalian expression system. And we will see that the choice of the expression system actually depends upon various things. For instance, what is the target protein property? Like, do you want to express a protein which is of large size or small size protein? For what application you need to express protein? Protein yield is also important, that if you want to do the structural analysis, you need to express protein with high yield, and you need to choose the expression system depending upon your need, like what will be the cost. If we use the bacterial expression system, it is inexpensive, and if we use the mammalian expression system, it is the most expensive system. Then what is the importance for your protein, that if your protein needs some post-translational modifications or not, because these post-translational modifications are not performed in bacterial expression system. So first of all, we will see the bacterial expression system. If we take an overview of bacterial expression system, we can see that first we have a recombinant plasmid, like we have cloned our gene Then we transform the plasmid into E. coli, and after transformation we grow the cells and induce the expression by using an inducer, which is IPTG. And after the protein expression is induced, we collect the bacterial pellet by centrifugation. Bacteria is actually the most common host for protein expression, and because it has the clear genetic background, its regulatory sequences which are involved in gene expression in E. coli and many other bacteria are actually well understood. The second advantage of using the bacterial expression system is the fast breeding, that it can be harnessed to express the cloned proteins at high level. We can get the high yield. Then the bacterial expression system is inexpensive. Bacteria are easy to store and grow in the laboratory, and they also need the inexpensive growth media. Then another advantage is high expression level. Bacteria can be grown in huge amount in commercial fermenters and provide the scientists with a rich source of the clone protein. And another advantage is that the purification of protein from bacteria is easy as compared to eukaryotic host system. Along with these advantages, there are certain disadvantages to use the bacteria as a host system, because if the formation of inclusion bodies occur, then it is hard to purify the inclusion bodies. For instance, this is the structure of the protein that you want, that it is your target protein and it is correctly folded into its proper three-dimensional structure. But when this protein is expressed in bacteria and the folding is not proper, means there is incorrect folding, it will result in aggregation of the proteins. Means eukaryotic proteins that you are expressing in the bacterial host, it will aggregate into insoluble cellular precipitates which are known as the inclusion bodies. And this happens because some of the heterologous protein do not fold correctly, and these proteins also do not undergo the covalent modification, or some of the proteins do not undergo the proteolytic cleavage. So because of this and many other reasons, some eukaryotic proteins are inactive when they are purified from bacteria, or it is not possible to express them at all. So these are shown the protein aggregates, which are actually the insoluble cellular precipitates, and we call them as inclusion bodies. So to address some of these problems, new bacterial host strains are regularly developed that include the enhancements such as the engineered strains contain the presence of the eukaryotic protein chaperone proteins or enzymes, and these chaperone proteins are involved in modifying the eukaryotic proteins. For instance, they are involved in correctly folding the proteins. Another disadvantage of bacterial host system is that imperfect post-translational modification occurs. As we know that if we compare the process of transcription in prokaryotes and eukaryotes, it differs, because in prokaryotes the genes which are involved in the same process, the genes are clustered together under the control of a single promoter, and we call them as operon. And when the messenger RNA is formed, the messenger RNA is known as the polycystonic messenger RNA, that it is encoding more than one kind of protein. And as the messenger RNA is synthesized, ribosomes become attached to it, and protein formation occurs, or the process of translation occurs. This is not the case in eukaryotes, because In eukaryotes, the DNA or chromosomes are located within the nucleus. The process of transcription occurs within the nucleus, and when the messenger RNA is formed, we call it as the nascent messenger RNA or pre-messenger RNA. Then the processing of this messenger RNA occurs, which involves the capping or attachment of poly-A tail, and then certain kind of post-translational modifications that occur, post-transcriptional modifications, and then it moves towards the cytosol. Within the cytosol, the ribosomes become attached to it, and then the protein synthesis starts, and which could be carried out by the free ribosomes, or ribosomes become attached to the ER, and then the proteins move towards the Golgi, and their glycosylation, phosphorylation, or other kinds of modifications occur. So the protein synthesis process is different in prokaryotes and eukaryotes because their systems are also different, and it is difficult to make the same eukaryotic protein in the prokaryotic host. So the major disadvantage is that when the protein when we express the foreign protein, eukaryotic protein, into the prokaryotic host, it will cause the imperfect post-translational modification, and the protein will have the low biological activity. And to overcome this problem, many specialized systems are being developed for expressing the protein in bacteria, and for this purpose, the promoter and regulatory sequences associated with the lac operon are often fused to the gene of interest to direct the process of transcription, and the cloned genes will be transcribed only when the lactose is added to the growth medium. But there is an issue of using the lac operon system, that the regulation in the lactose system is leaky, because we know that the lactose expression, or the genes which are involved in lactose metabolism, it is the expression of those genes is not turned off completely when lactose is absent, which is a potential problem if the product of the cloned gene is toxic to the host cell. And the transcription from the lac promoter is also not efficient enough for some applications. Another alternate system is also developed, which uses a promoter and RNA polymerase, which is found in a bacterial virus called the bacteriophage T7. If the cloned gene is fused to the T7 promoter, then it is not transcribed by the E. coli RNA polymerase, but instead it is transcribed by the T7 RNA polymerase, and the gene which encodes this T7 RNA polymerase, it is separately cloned into the same cell in a construct that affords a tight regulation, means it allows the controlled production of the T7 RNA polymerase, and this polymerase is also very efficient, and it directs the high level expression of most of the genes which are fused to the T7 promoter, and this promoter has been used to express the Rec-A protein in the bacterial cell. In this diagram, the regulated expression of Rec-A protein in the bacterial cell is shown. The gene encoding the Rec-A protein is actually fused to the bacteriophage T7 promoter, and then it is cloned into the expression vector. Under the normal conditions, which we can say that the uninduced condition, you can see there are many bacterial proteins but no Rec-A protein appears. When the expression of the T7 RNA protein or polymerase is induced in the cell, the Rec-A gene is expressed because the Rec-A gene is under the control of T7 RNA polymerase or T7 promoter. Then a large amount of a Rec-A protein is produced, and we can position the protein according to the ladder which is run along with the induced and uninduced sample. Please keep it in your mind that uninduced sample means when the expression of the T7 RNA polymerase is not induced, like bacterial polymerase is expressing its own, causing the transcription of its own proteins. And when we induce the expression of the T7 RNA polymerase, then the Rec-A gene, which is under the control of the T7 promoter, also shows its high level of expression. The second important system which is used to express the foreign protein or for heterologous protein expression is the yeast expression system. The yeast expression system is probably the best understood example because the yeast is the eukaryotic organism which is easiest to grow and manipulate in the laboratory. Like bacteria, this yeast can be grown on inexpensive media, and it is also ideal for large scale production of heterologous protein, and it is comfortable with genetic manipulation. Like if we want to make any genetic change in the chromosome of yeast, we can do it. And it exhibit near native conformation like post-translational modification and processing because it is also a eukaryotic organism. It has well-defined secretory pathways for extracellular export of the recombinant gene products, and it is usually safe to use. So this is the example of the vector which is used in the yeast or for yeast expression system. In the first step, you can see this is the multiple cloning site containing along with it the transcription termination. Here is the gene for His4 and here is the gene ampicillin resistance gene, and you can see the 5' AOX1 and 3' AOX1 sequences. These sequences are actually involved in the process of recombination because the gene of interest from the vector recombine within the chromosome or within the eukaryotic chromosome. So when we insert our gene of interest, here is the structure of the recombinant vector that it contains the 5' AOX1, then your gene of interest, and then termination of transcription of gene of interest, then His4 which is used for selection of recombinant yeast organism, and then 3' AOX1. When we linearize this vector, it will become like this. It contains the 5' P AOX, then gene of interest, His4 and 3' AOX. This is the AOX1 gene, and here the sequence is same at the 5' and 3' end, and then this plasmid sequence is integrated into the genome of the yeast organism. So actually the principle which is involved in the expression of the protein is the same as those in bacteria. Like cloned genes must be linked to promoter that can direct the high level expression in yeast, as we have cloned gene near the strong promoter of PAOX1. There are some disadvantages which are associated with the yeast expression system, one of which is that yeast have tough cell wall than bacteria, and it is difficult to introduce the DNA vector into the yeast cell. Just like in bacteria, when we want to transform the DNA vector into the bacteria, we use the phenomena of electroporation or heat shock. We provide bacteria some current or heat to make pores in its cell wall so that the DNA vector can move into the bacterial host. But in the yeast, the cell wall is tough as compared to bacteria, which make it difficult for the entry of DNA vector into the yeast cell. So for entry point of view, bacteria are more convenient for the genetic engineering and for maintaining the vector within the host system. Then yeast is a eukaryotic organism, but it belongs to the lower eukaryotic organism, and if we want to express some eukaryotic gene which belongs to higher eukaryotic organism, like we want to express the human gene or Arabidopsis thaliana gene, and that protein has some kind of post-translational modification which is of advanced type, not occurring in bacteria. So the foreign eukaryotic protein will not undergo the post-translational modification within the yeast because it lacks this specific enzymes or specific kind of mechanisms which are involved in post-translational modifications. Then another advantage of using the yeast is that it is much more expensive in comparison to the prokaryotic system. And some of the same problems that accompany the protein expression in bacteria also occur within the yeast. For instance, the heterologous proteins which need the proper folding, they do not fold properly because the yeast may lack some enzymes which are required to modify the protein to its active form, or the expression of the protein may become difficult by the certain features of the gene sequence itself. However, yeast is a eukaryotic organism, and the expression of eukaryotic gene is sometimes more efficient in this host than in bacteria, and folding and modification of the products may also be more accurate in yeast than for the proteins which are expressed in bacteria. The third most important system which is included in the eukaryotic expression system is insects and insect viruses. Insect cell system has the beneficial effect. It has the advantage that it has a similar post-translational modification that occurs in the mammals. So there is the similarity between the post-translational modifications of insects and mammals, and mostly the baculoviruses are the insect viruses which contain the double-stranded DNA genomes. When they infect their insect larval host, they act as a parasite and they kill the larvae and turning them into factories for virus production. And late in the infection process, the virus produces a large amount of two proteins which are P10 protein and polyhydrin protein, neither of which is required for the virus production in the cultured insect cell. So the genes for both of these proteins can be replaced with the gene of a heterologous gene protein. And when the resulting recombinant virus is used to infect the insect cell or larvae, the heterologous protein is often produced at high level, up to the 25% of the total protein present at the end of infection cycle. Baculovirus actually infect the invertebrates, and they occur in two forms, like single nucleocapsid, which are the virus particle and budding off from an infected cell, and second one is the polyhedron. Polyhedron is the cluster of these nucleocapsids which are known as the virion, and these virion are trapped in a protein matrix which is known as the polyhedron, and these polyhedron are released after the cell lysis. So on this slide, the two forms of baculoviruses are shown. This is the main structure of baculovirus, that it contains the cell membrane, and this is the nucleocapsid. And in the B figure you can see this is a virion, and virion is actually a cluster of nucleocapsids. That many nucleocapsids are clustered together, we will call it as the virion. Polyhedron is the protein matrix that you can see, it is in the gray color, it is the protein matrix. And polyhedron is the entire package of occluded virion. These are the different virions which are together. We call it as the occluded virion, and these virions are released into the environment after the cell lysis. When the cell lies, all of these virions will be released into the environment. So mostly the host insect cells which are used as a host are the Spodoptera frugiperda, which is also known as the SF or Bombyx mori. And the vector, which is mostly a baculovirus vector used, is Autographa californica multiple nuclear polyhedrosis virus. And promoter which is used is belong to the polyhedrin gene, because this promoter is exceptionally strong and it is used for the massive production or for high yield of heterologous protein. This is an example of expression within the insect host, or we can say that this is cloning using the baculovirus. As it is shown over here, this is the construction of a typical vector which is used for protein expression in baculovirus. The gene of interest is actually cloned into a plasmid between two sites which are known as the ATT sites. These are the sites which are actually recognized by the site-specific recombinase enzymes. So, gene of interest is inserted into the plasmid, and then the baculovirus DNA is mixed with it, and baculovirus DNA also contain the ATT sites which are recognized by the recombinase enzyme, and in between these ATT site it contains the DNA sequence which is required for plasmid maintenance in bacteria. And during the process of recombination, the gene of interest is inserted into the baculovirus DNA, and this DNA sequence, which is necessary for maintenance of plasmid in the bacteria, is recombined into the plasmid sequence. So after the recombination reaction is completed by the recombinase enzyme, now you can see that this is the baculovirus DNA, and this baculovirus DNA contain the ATT sites and gene of interest in between that site. And this is a small plasmid which now contain the DNA sequence which is required for plasmid maintenance in the bacterial host. Then the gene of interest containing baculovirus DNA is expressed after being transfected into the insect cell, and after transfection into the insect cell we harvest the recombinant baculovirus and transfect the infect cell for protein production. Here you can see that instead of using the term of transformation we are saying transfection. The term transfection is used when the DNA which is used for transformation, it includes the viral sequences and it leads towards the viral replication. Then we will call it as a transfection rather than transformation. In this diagram, it shows the insect larva which is infected with a recombinant baculovirus vector expressing a protein, and the protein expression shows the red color, and this shows the uninfected larva which was not transfected or infected with the recombinant baculovirus vector. There are certain advantages of using the insect as the host organism for expression of recombinant protein, because the recombinant protein which is produced in this host has complete biological function. It has the same post-translational modification mechanism that exists in the higher eukaryotes, and we obtain the high level of expression up to 50% of the total protein that is produced within the host cell is of the recombinant protein. And another advantage of it is that it can accommodate the large insert proteins, like we can clone and express the large size proteins into the insect host system. And another advantage which is very important is that we can simultaneously express the multiple genes in this host, and it is safer to use the insect viruses than mammalian viruses. But the major drawback of using the insect system is that it is not successful with all the types of proteins. The fourth system, which is used as a eukaryotic host system, is the mammalian cells in culture. We use the mammalian cells as a host for the production of heterologous proteins with a full complement of post-translational modification. And the most common mammalian cells which are used are CHO, which are the Chinese hamster ovary cells. And different types of cell lines are used for different purposes. Like if we want to express the protein for the short term, like we want the transient expression, then we use the cause DHK or the HEK293 cell lines. Cause is from African green monkey kidney, BHK is a baby hamster kidney, and HEK293 is human embryonic kidney. And if we want to express the gene for the stable system for long term, then we use the Chinese hamster ovary or CHO lines. And the vectors that we used in the mammalian cell, they are mostly the viruses, because it is the most convenient way to introduce the cloned genes into a mammalian cell by using the viruses. This method also takes advantage of the natural capacity of a virus to insert its DNA or RNA into the cell and sometimes into the cellular environment. And a variety of engineered mammalian viruses are available which can be used as vectors, including the human adenoviruses and retroviruses. The gene of interest is actually cloned, so its expression is controlled by the virus promoter. And virus actually uses its natural infection mechanism to introduce the recombinant genome into the cell where the cloned protein is expressed. And these systems have the advantage that proteins can be expressed either transiently or permanently, as we have seen in the previous slide. For mammalian cell system, we use the eukaryotic origin of replication. We can use the promoter, intron, multiple cloning site, polyadenylation site sequences, transcription terminator sequences. We use the intron because it enhances the production of the heterologous protein, and we also use the selectable marker gene, which is the SMG. And if we need to use the E. coli system with the vector of mammalian cell, then we use the E. coli origin of replication and ampicillin resistor gene. So mostly the vector which is used for expression in the mammalian cell system also contain the E. coli origin of replication and ampicillin resistance gene, also contain the eukaryotic origin of replication. It will contain the promoter sequence, intronic sequence, multiple cloning site, polyadenylation site, termination sequences, promoter of the selectable marker gene, polyadenylation site of the marker gene, and the transcription termination sequence for the marker gene. The major advantages of mammalian expression system are that it contains the same initiation signaling signals that occurs in the original organism. Then the protein processing and protein secretion and glycosylation and other type of modifications are same as that in the original organism, and the protein will make its native structure and it will keep the macromolecule intact. But the disadvantage is that the mammalian expression system is complicated and its requirements are very high, and the protein yield is low, and sometimes it also results in viral infection to the cell lines. So in the last we will do the comparison of the different expression systems with respect to the speed, yield, post-translational modification, cost effective, and its application. So the most efficient system that we can obtain the expression of the recombinant protein in less time with more speed is bacterial expression system. On the second number is the yeast expression system. Third is for insect expression system, and the speed for expression in the mammalian system is very low. For yield point of view, high yield of recombinant protein that we get is by using the yeast expression system. On the second is the bacterial expression system, then insect expression system, and there is low yield that we can get by using the mammalian expression system. Post-translational modification, if we see in relative to the humans, then the post-translational modification that mostly occur is the same as in the original organism we get in the mammalian expression system, then the insect expression system, then yeast expression system, and the least post-translational modification occur in the bacterial expression system. As the cost is concerned, bacterial expression system is most inexpensive, then the yeast expression system, insect expression system, and the most expensive is the mammalian expression system. We use the bacterial expression system if we want to purify the prokaryotic proteins or simple eukaryotic protein, but if we want some glycosylated protein expression or proteins involving the disulfide bonds or in intracellular or secreted protein, then we need to express our protein in the yeast expression system. If we want to purify the membrane protein or large size protein or viral vaccines or proteins which are involved in signaling pathway, cytokines or kinases, then we use the insect expression system. And if the protein of interest is complex eukaryotic protein and we need the more accurate post-translational modification, then we will use the mammalian expression system. So with this we are done with our today's lecture. In this lecture we have studied about the different host organisms or different host expression systems that can be used to express the recombinant protein, and after expression, so we can purify the recombinant protein which can be used for several applications. So for the expression systems, we started with the most simple expression system, which is the prokaryotic expression system or bacterial expression system. Then we move towards the eukaryotic expression system. Then in eukaryotic expression system, we have studied the three expression system. The simplest one is the yeast expression system, then insect expression system, and then we have studied the mammalian expression system. Then we did the comparison between all these four types of expression system, that each of these expression system has its own advantages and disadvantages, and we can choose the expression system according to the function of the protein, like according to the application for what purpose we want to express the protein in the host organism. This is all from my side. If you have any question, please feel free to ask. Thank you.